Now that we've considered the classes of membrane-associated proteins, let's look at some of their functions. We'll look at two broad classes of membrane proteins in terms of function. Transport proteins provide a means for substances to actually cross the lipid bilayer, while receptor proteins serve to communicate signals from outside to inside the cell without necessarily requiring membrane transport. We can classify membrane transport in terms of the energy cost associated with transport. Passive transport involves movement across the membrane without the expenditure of energy. In other words, the movement is spontaneous. Movement is driven by the presence of a concentration gradient, as illustrated below. A higher concentration of the substance pictured by the white circles exists on side 1 and a lower concentration on side 2. In simple diffusion, a molecule can move through the membrane without the need of a carrier protein. If we remember the hydrophobic nature of the lipid bilayer, we can see that substances that enter the cell in this way are most likely hydrophobic in nature. Since the concentration is higher on the left, the molecules diffuse through the membrane toward the right side, thereby increasing the entropy. We can express this mathematically by the equation shown. Notice that the equation predicts spontaneous movement toward side 2 only if the concentration on that side is less than that of side 1. This results in a ratio of concentrations, as given in the equation, that is less than 1. Since this is on a natural log scale, this gives a negative or favorable change in free energy. Another type of passive transport is referred to as facilitated diffusion. Movement is still dependent on a concentration gradient, but substances require carrier protein in order to move through the hydrophobic environment of the lipid bilayer. An excellent example is the glucose transporter carried by erythrocytes or red blood cells. Note that the direction of movement is always driven by the concentration gradient. Substances diffuse down the concentration gradient. If we compare the two types of passive transport, a difference may be observed in the rate of transport as a function of substrate concentration, as shown in the diagram here. In simple diffusion, illustrated by the lower curve, movement is driven solely by the concentration difference across the membrane. There is no saturation point. In contrast to this, notice the example of facilitated diffusion in the top curve. The plot is hyperbolic and saturates above a certain substrate concentration. Facilitated diffusion displays saturation kinetics due to the fact that there are a limited number of transporters. Once they are all saturated with ligand, they can't transport any faster, no matter how much more ligand or substrate is added. We will see examples of both simple and facilitated diffusion as we proceed.